Thank you. The floor is yours.
Okay, thank you very much, Sylvia. Nice to see you again, and I'm looking forward to meeting everyone else during the Q&A. As you know, I can't really see anyone now, so I'll be speaking into the void, which is always fun, but I'll do my best, and I'll basically read the paper. It was recently published as part of a focus section in the journal ISIS, so the History of Science journal. So the audience, the intended audience is really historians of science, and the paper is sort of an intervention in approaches to film and other media in within history of science, technology, and medicine. So I think I'll read the paper and leave room for further discussion about where it originated and where it might go from here for the Q&A. And thanks again for the kind invitation to the double joint seminar, and apologies again for the last minute cancellation or postponement last time. That was a very awkward time. So let's see. So the term intermediality is used by film and media scholars to evoke the continual dialogue between different forms of mass communication in a larger culture of production and consumption. For instance, the movie of the book, or the book of the movie. So this might be familiar to this term for those of you who are working more within film and media studies. It's encountered less frequently in histories of science, yet these two have examined relationally how a range of media have shaped the making and communication of knowledge. And this image is just, it's not something I'll talk about in detail. It's from a previous project, but just it's one of my favorite examples of a form of intermediality, a film strip of a obstetric model printed in a textbook. So multiple media kind of intersecting here in this image. So we have not only histories that focus on a particular medium, so the microscope slide, laboratory manual, X-ray photograph. You can think of other examples, I'm sure, but also explicitly or implicitly intermedial studies, notably of three-dimensional models and of moving images. And I'll just plug this upcoming virtual workshop that I'm not directly involved with, but it should be very exciting, coming up soon in November. So these studies have thrown into relief neglected aspects of the flat pages, which most historians are more familiar with. 19th century embryology is particularly well served by detailed intermedial studies of wax models. By comparing the production of models and books, these studies have expanded our view of scientific publication during the great age of print to include the other three-dimensional objects that became indispensable to university teaching. So printing, so like plastic publishing on wax, not only on paper. Wax, however, was marginalized by film and other media in the 20th century. By the mid-1950s, time-lapse was generally regarded as the perfect technique for the observation and recording of the slowly growing embryo. Rather like models, films of living developing embryos became indispensable for teaching and in some areas research. Footage was multivalent. Practitioners extracted individual frames for analysis and publication in print, and they spliced longer strips into films that also incorporated narration, still images, animated sequences, and other devices for rendering the raw material more intelligible. Histories of time-lapse methods in embryology have focused on the specificity of film as a temporal medium, as well as on the philosophical implications of digital imaging. So this talk takes a different approach, extending histories that have explored the production and uses of wax in relation to print and other media. It investigates time-lapse cinematography as a hybrid intermedial practice. To interrogate practices of authorship, publication, copying, storage, and especially distribution, it recovers the history of the embryonic development of Drosophila melanogaster, a film made by Eric C.A. Lucy at the University of Edinburgh in 1956. An unusually rich archive makes it possible to recover typically obscure uses and reuses of time-lapse footage in research, teaching, and other forms of communication. Conrad Waddington, this is a bit of background. So Conrad Waddington, an embryologist best known for the epigenetic landscape metaphor, a tool for visualizing developmental pathways, created the research film unit at the Institute of Animal Genetics, or IAG, in 1950. Waddington had joined the University of Edinburgh in 1947, and as director of the IAG, he incorporated film into a research program aimed at unifying genetics and embryology. He had first encountered the application of time-lapse to embryology in 1930 at the Strangeways Research Laboratory in Cambridge. There, he had used the watch glass technique to cultivate chick and duck embryos in vitro and collaborated with Ronald Canty, a pathologist and cinematographer, on a short film based on these experiments. Waddington had been impressed by Canty's films and the capacity of time-lapse to reveal otherwise imperceptible cellular dynamics. 20 years later, he established the film unit and hired Lucy, a recently graduated Bachelor of Science student with wartime experience in photography, to run it. And I should have asked Lucy's daughter, who's still around, what specifically his BSc was in and what kind of photography he was doing during the war, presumably some kind of espionage. I don't really know. Much like the Strangeways lab in the 1930s, the IAG film unit was an outlier. Universities in the US and West Germany increasingly boasted in-house film production facilities, but most British universities could not afford or were indifferent to them. Lucy, an inveterate tinkerer, had only modest resources at his disposal, as he worked mostly on his own in a large hut behind the institute. So this wasn't really a glamorous, film production studio or laboratory at all. He nevertheless kept filming there until he retired in 1989. His favorite camera for time-lapse was a Cine Kodak Special. With it, he filmed not only biological phenomena, but everything from crystallization to urban traffic. His best-known film, commissioned by the BBC in 1966, used a fast-tax, high-speed camera to capture the jump of a flea at thousands of frames per second. So this is also a period of, the democratization of 16mm film and these technologies becoming more accessible, including to scientists and filmmakers like Lucy. After Lucy's death, the University of Edinburgh's Centre for Research Collections inherited his film reels, audio tapes, video cassettes, photographs, and papers, and began the process, in collaboration with the Scottish Screen Archive and with funding from Wellcome, of cataloguing, preserving, digitizing, and publishing the films online. Lucy's daughter, Caroline Marr, donated further material, including letters, vinyl records, and photograph albums in 2013. 5 years later, in 2018, a selection of Lucy's films was screened with musical accompaniment as part of Timescapes, a series of public events hosted by the Edinburgh International Science Festival, a form of recycling that brought Lucy's life's work to entirely new audiences. In the 1950s, Lucy's films were seen by a select few, but a film, and there's comparisons, I think, here with aesthetic and institutional comparisons we might be able to make with the avant-garde, and we can pick that up in the Q&A perhaps. But a film he completed in 1956 on the embryonic development of the fruit fly would circulate widely in the 60s and 70s. So the Drosophila embryo, the fruit fly embryo, was a marginal experimental object prior to the 1970s. So for those of you who aren't steeped in the history of biomedicine, we typically think of the fruit fly and the mouse, the zebrafish, and a handful of other organisms as the kind of key research material or experimental systems in laboratory experimental biomedicine. So, and that's for the fruit fly. In the case of the fruit fly, that's sort of where so much genetics research was done in the first half of the 20th century. We're talking really about the adults. The eggs were basically ignored, except as just a means of producing more adult flies. From 1910 to 1940, Thomas Hunt Morgan and his students established the adult fly as a key organism in genetics research. The Morgan School, which moved from Columbia University to Caltech in 1928, became the center of an international network of Drosophilists with a shared experimental culture and toolkit that included standardized mutant strains. And there's this wonderful book by Robert Kohler, Lords of the Flies, which is all about this. Drosophila reproduced more quickly and prodigiously than other laboratory animals. and crucially all year round, so you don't have to wait for the kind of seasonal breeding season to do experiments with it. The adult was especially amenable to manipulation, but in a move that reinforced the separation from embryology, between embryology and genetics, the fly's tiny, fragile, and opaque eggs were generally excluded from research. Hermann Mueller, a former colleague of Morgan's at Columbia, introduced mutant strains via the USSR to the IAG, to Edinburgh, in 1937. Mueller returned to the US in 1940, but the refugee geneticist Charlotte Auerbach, pictured here, maintained his stocks through the war, mutagenizing them with mustard gas. And after the war, Waddington had first worked on Drosophila while visiting Morgan's lab in 1938-39. As head of the IAG in the 1950s, he diverted surplus eggs from Auerbach's stocks for research that compared normal and mutant embryos. So basically, they're doing this research in Edinburgh on the adult flies, and they have these eggs anyway. And Waddington, who's always had this sort of desire to merge, to integrate embryology and genetics, decides to hive off some of the eggs and give them to his PhD students for research on embryological genetics, which is comparatively niche at the time. Such comparisons, so between normal and mutant eggs, were facilitated, so this is the early 50s we're in, were facilitated by the recent publication in 1950 of Demerich's Biology of Drosophila, pictured here, the first book to synthesize information about the fly's embryonic development and adult anatomy. So this new resource had just become generally available. Crucially, the chapter contributed by Yale's Donald Poulsen used photomicrographs of living dechorionated eggs to stage normal development in Drosophila, and in so doing, prepared the way for experimental work on its developmental genetics. So those pictures on the side are of living eggs where the opaque shell has been removed to actually see what's going on inside under a microscope. So this isn't cinematography. This is photomicrography, or basically photography through a microscope. In the early 50s, and this is sort of like not the glamorous experimental work that most of us think about, but this is basically the foundation for embryological research, are these developmental stages that are produced for sort of for every species. In the early 1950s, Waddington tasked two doctoral students, Sheila Counts and Donald Eade, with investigating the embryos of mutant strains that did not survive into adulthood, so these lethal mutants. And the idea is that there's something fundamentally, like the mutation is because it kills the embryo, it must be like the thing that's being mutated must be pretty crucial if the mutation results in death. So that makes it kind of interesting to study. He assigned Lucy to their projects, so Lucy was assigned to film for the PhD students abnormal development in mutant embryos. Equipped as they were with Poulsen's, they already had Poulsen's photographs of normal stages, so they didn't need Lucy to film normally developing embryos for them. But crucially, because it was impossible to tell whether any particular egg set up for photography was of a normal or mutant embryo until development was underway, Lucy ended up filming normally developing embryos alongside the mutant ones. And here in the primary source material, the terms, like when it comes to time-lapse, one of the things I noticed, others, you may have noticed this too, is photography and cinematography are kind of used interchangeably at the time. So it's basically you set up a camera and you take these photographs every a few minutes, and you end up with a reel of footage. But oftentimes the purpose isn't to actually make a projectable edited film. The idea is just to be able to extract individual key frames from that footage. So that's something else I'd like to discuss a bit more in the Q&A. Lucy ended up filming normally developing embryos alongside the mutant embryos. Eade and Counts, the PhD students, analyzed stills from the time-lapse footage for their PhD thesis. So this was like raw, visual evidence that was integrated directly into their PhD work. And Lucy included sequences in the film he made that was completed in 1956, a 19-minute black and white sound film that also incorporated narration, diagrams, and apparently poorly focused photographs of microscope sections. And here are just some images you can see from one of the PhD theses and from a published article that I'm about to discuss. Ede and Counts further used Lucy's footage as the basis of a research article in Wilhelm Roos' Archiv für Entwicklungsmechanik, a journal of experimental embryology. Time-lapse, they argued, facilitated new observations. And this is I guess the crucial intervention of the article. One of the things I want to do with the paper this talk is based on is give an example, elaborate an example of film being used in the context of laboratory research in biomedical research to produce scientific knowledge in a way that was complementary to to microscopy, photography, and other kind of research tools. So the use of the cine camera very much as a research tool, not only for teaching or sort of communication. So time-lapse, they argued in the paper, facilitated new observations on those phases of development which involved large rearrangements of the embryonic material, in particular on blastoderm formation, gastrulation, and involution of the head. So you can imagine sort of and now you can watch these kinds of videos on YouTube, but basically the cells kind of migrating around in these very big movements of cellular material that became visible to them using the time-lapse technique. More precisely, they reported aspects of gastrulation, so that's the formation of the gut, that had not been described elsewhere, but were well shown in the film. They also revised the account of mouth formation that Poulsen had advanced in Biology of Drosophila, the book I mentioned. Their claims seem to have been broadly accepted. So I'm not a Drosophila embryology expert by any means, and so it's difficult coming, from a history background to evaluate all the claims made in this kind of context. But I haven't found, I didn't find any evidence of resistance to the article, of pushback, or of controversy. The claims, if not earth-shattering, seem to have been broadly accepted and encouraged dozens of similar cinematographic studies. And the paper, the 1956 paper, the cinematographic study of the Drosophila embryo, would be favorably cited for decades to come. Lucy received little credit. Ede and Counts thanked him in their 1956 paper as they might have a lab technician. So there's kind of an acknowledgement for him, but they stopped short of including him as a co-author. Catalogs and lists typically identified the Drosophila film, not with Lucy, but with Ede and Counts as the authors, or just Ede. This was par for the course, with the exception a decade later of the classic Flea Jump paper on which Lucy appeared as second author. Lucy labored in obscurity. So film has been, Hannah Landecker described film in ISIS, the journal, as an infinitely, quote unquote, infinitely replicable and widely distributable medium that circulates easily across many cultural domains. So this may be true today. It may just be a kind of exaggeration. It may be true today in the digital era, but copying, distribution, and circulation were not always so effortless. And these are kind of the aspects of film history beyond kind of visual analysis of representation that I really wanted to kind of push at with this paper. So copying, distribution, and circulation. In the recent past, supply was severely constrained by the high cost and material limitations of physical media, and by the lack of distributional infrastructure. Even today's sprawling entertainment film industries had first to establish and scale up their own systems of copying, storage, and mass distribution that are like very easy to take for granted, because they're sort of invisible and seamless, I guess, except when cinemas stop functioning because of a pandemic or something. Laboring on a smaller scale and without the benefit of lucrative commercial markets or promotional apparatus, universities and other institutions cultivated alternative networks for niche products. In the UK, distribution for scientific films was centralized only with the creation of the Higher Education Film Library in 1972. So I think these film libraries, these distributional infrastructures, as I call them, are a really important part of the story. I mean, without them, Lucy might have made this film, but how would anybody get to see it? It wouldn't really be able to circulate. At first, Lucy was, in fact, the sole distributor of his film. In a footnote to the 1956 paper, Eden Counts explained how to obtain a copy directly from him. So like you write a letter to him and ask for a copy, and the address was given in a footnote to the paper. From 1959, German university instructors could also rent a copy from the Institute for Scientific Film, the IWF, in Göttingen. This was like a massive, large-scale project to centralize scientific film production and distribution in Germany, in West Germany, after the war. Lucy's correspondence and other sources indicate that the film circulated widely and was much valued as a teaching aid in the 1960s and 70s. Methods in Developmental Biology, a landmark American laboratory manual from 1967, endorsed it as a valuable tool in visualizing dynamic aspects of early development. And a review in the British Journal of Biological Education recommended it as an excellent record of insect development suitable for advanced classes. Poulsen's copy suffered so much wear and tear that he stopped lending it out, and decades of use and teaching left other copies similarly battered. And I was able to kind of find that out because of the somewhat unusual archive at the University of Edinburgh that contained correspondence that we can discuss further in the Q&A. And that's one of the reasons I picked this film to work on, because there was an archive available for it, unlike most films that I would like to work on, but for which no kind of complementary paper archive exists. So all in all, Lucy's film had an extraordinarily good run. As late as 1980, it was still in constant demand for teaching purposes in the UK and overseas. At nearly 25, however, it no longer represented the state of the art in embryology or imaging. The film had become dated epistemically and technically. So when Mary Bounds, a young developmental biologist who had recently restaged the fruit fly egg using newer microscope technology, joined Lucy at the University of Edinburgh in '79. He decided to make a remake with her help. Bounds spent months supplying Lucy with living eggs for filming, but it seems likely that he never completed the project. The original 1956 version remained in circulation well into the 1980s, and Bounds used a silent rough cut for her teaching. So she would just project unedited sequences and talk over them when she was giving her lectures. And this is just, I didn't really get into the remake very much in my published paper, and the remake is what generated the paper archive. And this is just this is just a map of the campus where kind of the route Mary Bounds would have taken, carrying the living fruit fly eggs across to where Lucy's film studio is based at high speed, not spilling them and trying to keep them warm on her overalls so that they wouldn't freeze to death. Again, this is I haven't really done anything with this yet. You can talk more about sort of what the possibilities of what to do with this kind of material. This is these are pages from Lucy's kind of a working script, as you could see, very much a working script, a work in progress in terms of how he envisioned the final film. So in this case, really intending to make a film for teaching purposes, unlike the original film in 1956, that was kind of a accidental byproduct of filming that was being done for the PhD projects. So research labs increasingly produced their own time-lapse sequences or on cheaper, more convenient videotape for private in-house use in the 80s. Again, and most of these were not up to publication standard and were not really intended for publication or teaching or circulation. They were just sort of for private use within a particular research lab. But became increasingly possible and cheap and practical for scientists to kind of make their own videos, and that's what they did in the 80s. In terms of reach, Lucy's Drosophila film may not have been superseded until the publication in 1991 of A Dozen Eggs, a collection of embryological time-lapse videos on VHS that was sponsored by the U.S. Society for Developmental Biology as part of an initiative to broaden access to teaching material. So again, this is predominantly this is film as a kind of teaching aid. A Dozen Eggs was distributed by Cinauer, who published the leading textbook in the field and was sort of branching out into multimedia at the time. It was edited by Rachel Fink, who's pictured here. although I couldn't find this is a more recent picture. So a developmental biologist at Mount Holyoke College in the US, who also made the film on fruit fly embryogenesis. For years, Fink had collected, copied, and mailed out tapes of the most teachable time-lapse sequences. So she kind of was at the center of an informal network of video distribution through the 80s. A Dozen Eggs consolidated her previously informal activities. It would be cataloged, reviewed, purchased by libraries, screened in laboratories around the world, and published on DVD in 2008, evidence of its enduring relevance into the digital 21st century, presumably until YouTube comes along and sort of replaces these, basically physical video and digital media. Lucy and his film, meanwhile, faded into obscurity, like I think fairly complete obscurity, it's probably safe to say, until preservation, digitization, public exhibition, and online publication, all forms of reuse and recycling, which was the theme of the ISIS focus section, breathed new life into his works. So in conclusion, extending histories of embryological media from print and models to films, this talk has approached time-lapse cinematography as a hybrid intermedial practice. So instead of trying to isolate what's sort of specific about film as a medium, I really wanted to integrate it alongside all the other adjacent media and show how it was sort of inextricably wrapped up, how film production was also part of microscopy, photography, etc. Doing so has brought into focus sort of significant entanglements between film, photography, print media, also books, theses, journal articles. It's also integrated, this talk is also integrated practices of authorship and publishing, as well as copying, storage, and distribution, crucial yet neglected infrastructural activities that have made viewing possible on a mass scale. As we've seen, the lack of distributional infrastructure significantly limited the reach of scientific film until the creation of specialized library services such as the IWF in the second half of the 20th century. Starting in the 80s, first videotape and then a succession of digital technologies widened access to production and consumption more than film ever could. Today, moving images are ubiquitous in research, teaching, and other forms of communication. Like models, and that's part of the, I guess, that's partly responsible for the interest in the history of them, including on the side of scientists. Like models, like wax models, they should be included alongside print and other media in histories of scientific knowledge. Thank you.
Thank you, Jesse. Thanks. That was an amazing